Okay. So, I'm gonna record. I'm gonna actually abuse you guys for a few minutes. I hope you don't mind, because I'm really nice when I abuse people. So listen. Here's the situation. I decided, just for kicks, that I would see who read the recitation paper for today. I can tell, and it was really scary. The number of people who didn't even open the paper, and I'm looking around and I see a lot of those people aren't here. But it makes me kind of feel like, well, what's the point, right? Yeah.
To be fair, some people don't have
No, I'm talking about the people for today.
Oh.
Yeah, I would never. If you're on Friday, God, I would never expect you to read the paper today. God, you're insane. Don't you have other things to do with your life, like eat? No, people for today. So it's just really kind of discouraging on my part because I really want everyone to do well. And then when I see people who don't even open up Blackboard to look at the paper, I'm like, well, why are you here? Why are you here? And then I start getting all existential. And then I start thinking about my PhD advisor, who I may have mentioned. He was really abusive, but it was probably a good thing in the end because, well, we get along great now. But he used to motivate me by telling me I would fail. And it didn't work really well because I wasn't motivated by that. But I'm just going to let you guys know, if you don't work hard, you will fail. Now, I was working hard, and he would tell me that I would fail. And that I just thought he had unrealistic. He would say things like, well, if you weren't, going out and eating, you would do better. And I was like, well, we need to eat. And we weren't doing badly. But I just want everyone to understand that you have to put the time in to do well. So a lot of you guys are, and you guys are doing really well, and that's awesome. But for those who are feeling challenged and are not passing anything, opening Blackboard is a good start to doing well. And if you just are like bitter about being here, then think about, why are you doing this to yourself? Why put yourself through misery for nothing, right? Anyways, okay, I'm done with the abuse. Was that horrible? No one's crying. Are you guys going to go to your next class and talk about me? Dr. Ehrlich went nuts today. Okay, so we've been talking about the cytoskeleton, and this is what we've done so far. So we've been talking about the components: actin, microtubules, intermediate filaments. Uh-oh. Who's looking sad? Are we looking sad? Oh, there's a wasp. Is it sitting on anyone? Wasps love to come into this building. They're always in here. Okay, who was in my class? Who's in recitation and just picked up the wasp? Was it you? No.
It wasn't me, but I was there.
You were there. Someone just picked up the wasp and put it out. Where is that person? She's not here? Okay, bummer. Anyways, we have a very brave person in this class who's not here. Okay, so let's continue. Sorry about the wasp. If I brought wasp spray to class, that would probably be worse even, because then you'd get all sprayed with. Okay, so we're talking about the cytoskeleton and how it's all about monomers coming together to make polymers, and that's all involved with these major concepts of nucleation, critical concentration, plus and minus ends, scheduling, and dynamic instability, which hopefully now, after a few days, you guys all understand those major concepts very well. And the assembly and disassembly is regulated by NTP hydrolysis. So on Friday, we started talking about proteins that nucleate. By nucleate, that means they put those first few monomers together to form a nucleus, and that allows growth. So a protein that nucleates is always going to encourage growth of polymer. And we started with talking about gamma tubulin ring complex and Merco tubules, and how it nucleates microtubules. Just ignore my spelling errors. Okay, so this is where we ended. So, in animals, a microtubule organizing center located near the nucleus, called the centrosome, contains lots and lots of these, more than 50, gamma-tubulin ring complexes. So these are these nucleating sites that the microtubules grow out of. And it looks like this, and it's all these gamma-tubulin ring complexes, and the microtubules grow up and out, and that's how you get that star kind of appearance. And they're pushing outwards against the plasma membrane, and that's why your centrosome is centered, right? You can kind of visualize that. If everything keeps pushing out, the thing that's in the middle is going to be in the center, because the cell is pretty regular in shape. Centrosomes duplicate during interphase. No, not interphase. Prophase. And move to the opposite sides of the nucleus to form the mitotic spindle. See, there you go. They're your microtubules growing from the gamma-tubulin ring complexes of the centrosome. So I just wanted you guys to, I love immunofluorescence. So every time I think something will look nice fluorescent, I put it up. So this is showing, so ignore like this masking thing. This is just from a paper where they were looking at this protein masking and they were trying to show it localized to the centrosome. I don't care about that. Ignore that. I want you guys to, it's just like a nice marker for looking at the centrosome. And so I'm putting this up here so you can see how microtubule organization changes through the cell cycle, right? So over here, you've got, here's your DNA, which is blue. Let me turn off the light. Right, so you've got your DNA, blue, and then your microtubules are green, and they actually have their centrosomes red, and the reason they look yellow is because, for whatever reason, red and green overlay make yellow, so that's usually how you tell co-localization, but don't worry about that. So you can think of your centrosomes here. So here you've got one little dot of yellow, so that's one centrosome, microtubules radiating out from the center, right? And then as you start to go through mitosis, I thought I had that labeled. Okay, as you start to go through mitosis, you get centrosome duplication. See, there's two. And then they move outwards, right? So here's your mitotic spindle. And now you can watch your DNA. It's lining up. What phase of mitosis is this? Metaphase, right? And then you start to get, they're attaching, the spindles attaching to the chromosomes and separating them, and then you get cytokinesis, and you have two separate cells. So you can learn a lot about what phase of the cell cycle your cell's in, how close it is to mitosis, whether it's in mitosis, just by looking at your centrosomes, because they only duplicate when they're going to form the mitotic spindle. And then one thing I just wanted to kind of put in your heads for now is that centrosome duplication is regulated by phosphorylation by the Aurora A kinase. We haven't talked about it yet, but in a few weeks, as we start to talk about cell cycle, we will. And so you always want to be thinking to yourself, she's talking about centrosome duplication. What regulates centrosome duplication? It just magically happens. And someone will say mitosis. Well, what turns on mitosis? So there's always something, some protein is being phosphorylated or being degraded or something like that, and that's going to regulate whether different proteins can interact. And Aurora A is a kinase that is involved in regulating centrosome duplication. And just remember that for now, because it all builds on itself, and especially when we talk about microtubules and then we go into cell cycle. It's very much related. Okay, I think I have more. Okay, so here's a practice problem for you guys early on in class. You didn't have to wait till the end. Okay, so you're studying cell cycle regulation. You're testing different drugs to identify those that cause cell cycle arrest at early mitosis, so somewhere around prophase, metaphase. And your experimental conditions are treat cells in culture with drugs for 12 hours, and then you assess the drug activity through IFA, immunofluorescence assay, using antibodies against gamma-tubulin ring complex and DAPI to visualize DNA. Here's the result of the IFA after treatment with compound A. Was your drug active? Why do you think so? What results do you expect if your drug is active or not active? So I want you guys to talk to your friends for a minute. Look at this. And right now you might be going like, wait a minute, we're not talking about cell cycle. What is she talking about? Hopefully you guys can put it all together and understand why I'm asking you about cell cycle and gamma tubulin ring complex. And talk about it, and we will reconvene in like 2 or 3 minutes, okay? Oh, right, guys, sorry. I just heard a question that, so the gamma tubulin ring complex is green, and DAPI is blue. DAPI, DNA is blue. Okay.
So, in all of them, you see the formation of two ring complexes, which tells you the mitotic spindle is forming. And that's basically what happens in the left phase. It looks like a spider edge, so it looks like everything's proceeding. So the drug would be
Got me to...
I mean, there's some that look like, you know, they're not doing that, but
There's a lot of quiet. Does anyone need clarification on the question? Yeah. Do you have a specific question? Okay, so I'm really asking you, you're working, like, let's say you're working in a cancer lab and you're doing drug discovery and you want to find cells that cause cell cycle arrest. Arrest of the cell cycle means the cell cycle is not moving forward and it's stuck at early mitosis, which means prophase or metaphase. Okay, so you know what cells should look like if they're in, let's say, interphase versus mitosis, and I hope you know. And you know that gamma tubulin ring complex is a marker for something that changes. And if your cells are resting, then they should all be looking the same. Now that's, I really gave it away. Okay, so try talking again and seeing if you can understand this.
if it was
Okay, guys, do you want to talk, or are you guys deep in deliberation? Okay, ready? So someone tell me, was the drug active or not? Yes.
You think it's active mostly because in some of the cases you can see like it's stuck, like the bottom two, you can see it's stuck in metaphase.
Uh-huh.
And then the top two at this side, you can see some centrosomes haven't duplicated at all, so it's probably still stuck in prophase.
Okay. So let me clarify my question. If your drug is active and it causes cell cycle arrest at prophase/metaphase, all of your cells should look the same, because imagine the cell cycle's moving forward and then it hits a roadblock. So they're all going to keep moving forward till they hit the roadblock, and they're all going to look the same. So I see what you're saying, and I think me putting prophase and metaphase confused you guys. So I want you to just think of it as early mitosis and concentrate on the staining with the antibodies against the gamma tubulin ring complex more than on the DAPI for the DNA. Because, yeah, you're absolutely right. You've got some metaphase. You've got some metaphase cells here, but here you don't. Right? So, anyone else want to tell me what they came up with? Anyone think that the drug is not active? Okay. Does anyone want to share the reasons why?
There is duplication of the insert, and when there's duplication, well, the centrosomes, when there's duplication of it, then that means it's preparing to form the mitotic spindle mitosis. So,
Okay, so you think it's not active because of centrosome duplication. Well, so the other information that might be helpful is that centrosome duplication happens at early mitosis. It happens at like prophase, metaphase. So if you're going to have a roadblock put there, some kind of stopping of the cell cycle, you will see. You can see that. So, yeah.
I said it was not active because I saw it forming like little spindle things, like it was getting ready to fall apart. So then I thought that I was going from metaphase to anaphase, and so I said it was active.
Okay, well, that's actually good logic. Yes, yes, yes. You all have very good logic, and I'm realizing my question was poorly written. So, okay, so I'm hearing lots of things. What Ashley said is that here you're seeing almost anaphase, right? Because you have metaphase, but then you have the, here you almost have the pulling apart of the chromosomes. But really what I want you to focus on is like, what's going on here? Where is the cell in mitosis? Is it in mitosis? Nowhere near mitosis, because how many centrosomes are there? One. How many are here? How many are here? Two. Well, one's kind of over here. What about here? Okay, so would you say that the cell cycle is being arrested in this culture of cells? Mind you, these are in a plate in your incubator all together. They're not taken from different places. And they were all treated with the same drug. So they should all keep going until they hit a stopping point if the drug is working. And so because you see cells that are in interphase, you see cells that look like they're in metaphase, you see cells that look like they're in anaphase, you see cells that look like they're in prophase. There's no cell cycle arrest because they're all in different places. And so frequently, when you're studying things in cells, if it has anything to do with cell cycle, you'll synchronize your cells. And the way you synchronize them is by putting a drug in that's going to arrest them at a certain point. And so they'll all kind of move, and then they'll stop at, prophase or metaphase. And then you'll take the drug off, and then you'll let them go again. And then they'll all go through cell cycle at the same time. But so cells, remember, in culture are all going through cycle at different times. And that's why you're seeing cells in interphase and prophase, interphase, prophase, metaphase, and anaphase all in the same picture. But the drug is not working. Okay, so if your drug is active, based on what I just told you, what would you expect to see? All cells are at the same place, and all cells are probably like here. So if your drug's not active, it would look like this. So why are we talking about cell cycle here? Like, what does this have to do with the cytoskeleton? Right, so cytoskeleton, so the centrosome duplicates to form the mitotic spindle, right? Mm-hmm. So, okay. That didn't go nearly as well as I was hoping. But that's okay. I think it required much more prior knowledge than I anticipated. But do you guys understand what I was trying to do? Okay. Does anyone have any questions? Is anyone like, I have no idea what you're talking about, and I want to cry? Who's crying? No one. I didn't bring any tissues, so that's good. Okay. Good. I try not to make people cry. Okay, so centrosome. This is kind of what I was saying before. The centrosome during interphase is going to be one centrosome with the microtubules radiating out, and it's pushing against the sides of the cell, and that's what causes it to be in the center. And this is an experiment where they took a centrosome and they put it in a plastic well, so just like in some kind of tissue culture dish, and put tubulin subunits, and so they put the centrosome here, and over time it's centered, and that's because you have the gamma tubulin ring complexes, and they're gonna nucleate the microtubules, which are gonna radiate out, and as they radiate out and push against the edges of the wells, it ends up in the center. Hence the centrosome. Okay, and so part of the reasoning behind the centering behavior of the centrosome is it's involved with positioning of organelles in the cell. Because remember, microtubules are involved in movement of things. It's not just about mitosis. They're involved in movement of vesicles, movement of organelles. Okay. Oh, God, there's a lack of pictures recently. Okay, so microtubule nucleation, it occurs in the center of the cell, right? You guys all agree with that? Yeah. Why does microtubule nucleation occur at the center of the cell? Huh? Yeah, I hear someone going, centrosome. Okay, thank you. Centrosome. Yeah, because that's where your gamma tubulin ring complexes are. Okay, so based on the function of actin in cell shape and movement, where would you expect actin nucleation to occur? Where do you think actin nucleation would occur? Should it also be in the center of the cell? Near the cell membrane. Yep, near the cell membrane. Absolutely. Okay, so it occurs at or near the plasma membrane, just beneath, actually, the plasma membrane at the cell cortex, and it determines the shape and movement of the cell surface. So this is involved with formation of microvilli, filopodia, lamellipodia, and phagocytic cups in macrophages, right? So anytime you see the plasma membrane changing shape, you're gonna have actin polymerization, and so there's going to be some kind of nucleating protein there. And this is often regulated by external signals. That was the example we had early on of the macrophage, or no, neutrophil chasing the bacteria, right? So there was an external signal. There was some kind of chemoattractant being released by the bacteria, and then the neutrophil was chasing it, right? And that requires a lot of actin nucleation, correct? Yes. Does actin have anything to do with movement? Oh, God. I was in the ER like all day yesterday. Totally sucked. My son, they had to like stick a thing down his nose, and I'm here. I'm here, kind of prepared. I'm prepared. I got a talk to give. Put some new pictures in it. Yeah, he was breathing like this. That's not good, right? So, and he didn't have a voice anymore. I don't know what happened. It just went away. So we took him, called the doctor, and he's like, you should take him to the ER. So I took him, and they kept us there from 2 till 8 because I was like, should I go to Hopkins or should I go to GBMC? And I was like, well, I don't feel like going to Hopkins. It's such a pain in the butt. So I went to GBMC. And then they had to get the doctor to come from Hopkins. Anyways, I was like, oh, I guess I should have gone to Hopkins. They actually had to call an ENT specialist to come from Hopkins. I was like, you people. Anyways, I'm here. Okay, so actin filament nucleation. So we're talking about formation of actin filaments so that cells can move in some way. Now you need to be thinking, well, what's nucleating actin, right? Right? So there's the ARP complex, and then there are formins. So the ARP complex is very similar in function to gamma tubulin ring complex. It nucleates the filament growth from the minus end, allowing elongation from the plus end. So it binds to one end, and you have assembly of monomer at the plus end and growth. So this is how it looks. It also likes it. The efficiency of ARP2/3 is much higher when it binds to actin. So usually, when it binds to actin filament. So usually, what you'll have is you'll have an actin filament formed, and then ARP will bind to the side of it and form, and then add on monomers, and it will grow like that. See, so you've got, and it results in formation of an actin web. This is quite different from formin, so we'll talk about that in just a minute. So the structures of ARP2 and 3 are quite similar to actin. That's not too hard. That's not too hard to imagine if you think about it for a minute, because it's nucleating. It nucleates actin filaments. So that means it's going to, remember, so actin is binding to actin, head to tail, right? So it's logical that whatever's going to start it is going to maybe want to look like actin, because actin likes to bind to actin. And so here's a structure of actin, and here's a structure of ARP, and its plus ends, which is where the subunits get added, look quite similar, right? So ARP2, ARP3, and they all both have this kind of binding site for where the that minus end of the next actin monomer is going to come in. Okay. So here is how it looks. You'll have an inactive ARP complex. Something will activate it, right? Because obviously all this is regulated. If you have ARP complexes that are always active, is that good? No, right? Because you only want actin polymerization at certain places under certain conditions. Does that make sense? Do you want just phagocytic cups forming all the time for no apparent reason? No. So you'll have some kind of activating factor that's going to come, and it's going to activate the ARP complex, and that's what allows addition of actin monomers, and then you have addition of the monomers to the plus end, and you have the filament growth. And it forms this web like I just told you about, right? So the complex nucleates most efficiently when bound to the side of another actin filament. So you'll start with ARP2/3 that's gonna form a complex, and then you'll have another one will bind, and then it'll look like this, and it creates this web-like structure of actin filaments. And this is associated in animals, at least, with structures at the leading edge of migrating cells. So if you were to, well, I think I have a picture coming right up. There you go. Okay, so here's another little practice problem that we can do together. So you're studying the ARP complex, and you want to know whether it's involved in formation of the leading edge. Leading edge is the part of the cell that's pushing forward when it's moving, right? And you have fluorescently labeled antibodies against actin and against ARP3. So you do immunofluorescence, and you see the image below. Actin is red. Let's turn off the light. ARP is green, and remember I told you when red and green overlap, you get yellow. Okay? So where would you say the leading edge of the cell is? Top, bottom, left, right, east, west, north, south. Let's do north, south, east, west. North, right. See all this yellow here? So here's there's a lot of random red, there's a lot of random green, right? Suggesting that a lot of inactive ARP is kind of just hanging out in the cell, and a lot of actin is doing other things too. But look at this really bright yellow, right? So you've got ARP and actin being recruited, and so probably there's some kind of chemoattractant up here, which is causing the cell to want to move that way. Okay. So you can make the assumption that if you were looking for nucleating proteins at the leading edge of a cell, it would probably be in an ARP kind of nucleating protein. Let's leave that on. That's better. So you don't like that one?
No, I figured it was
You don't like the leading edge? That's what happens in this room is that you have, like, you can't leave, right? You cannot leave without me talking to you. There's no way. I'm like the gate master. Okay, here's a question from a quiz. The ARP complex controls the nucleation of actin filaments in response to extracellular signals by what? If you have your letters, you can hold them up, or you can do like, I don't know, that was like an F. There's no F. Okay, I see one person with a D, another person with a D, another person with an A, a P? Oh, a D, a D. We got a human D. Okay, so D's and A's. Are we ready to duke it out? What do we see up there? D, another D. D's and A's. Yeah, okay, so we're going to have to duke out the D's and A's. Okay, so what does ARP do? We just said it will nucleate actin filament growth at the minus end, allowing for rapid growth at the plus end, right? Yep. Connecting different actin filaments, right? Because it binds to the side and nucleates, so you get that web. Yep. Nucleating actin filaments at the leading edge of migrating cells. Yeah, it does that. So that'd be D. That'd be D. So good job, those who participated. Okay. So right. So just something to think about. Gamma, tubulin, and ARP proteins, they are both two conserved nucleating proteins, and they're very similar in structure to tubulin and actin, right? So we just kind of talked about that. So you could think about, like, well, where did they come from? They're so similar. So they might have, they may have arose from gene duplication, and then they acquired some kind of nucleating properties, and so they were maintained in the genome. So there you go. So the ARP complex forms these branched web-like actin networks. And so now we're going to talk about the other protein that nucleates actin, formins. And they form parallel bundles of unbranched, so no branching here. No branching, formins. Unbranched actin filaments. And so they're involved in things that are more like you can more think about as contractile. So such as a cleavage furrow, right, your contractile ring in the dividing cell, and bud growth in yeast, and that's also very similar, where you have that pinching off. So those are two very different functions. So if you're ever thinking about movement in terms of a leading edge, you should be thinking about ARP complex. And if you're ever thinking about anything that's contractile, then you should be thinking more about formins. Question? Answer? Anyone? No? Everyone's happy? Good. Does anyone want to hear about what it was like to be at the ER yesterday while they shoved a camera up my son's nose? It was fine. They actually had to wrap him in a sheet because he's 2, and it was like, I didn't like it at all. No one likes that, right? Who wants a camera shoved up their nose?
Was he okay?
He's fine. He's fine.
What was wrong?
He was breathing funny, and he has croup. That was it. But they took an X-ray to make sure he didn't swallow something, because at 2, babies would be like, "Hey, what's this? Let me shove this up my nose," and they saw something in the X-ray. So it's like this huge thing, but they never found out what that thing was. And then they're like, "Well, we think it may have been artifact." Well, thanks. Anyways, okay, formin. Formin dimers bind actin and encourage nucleation by capturing monomers. So it's totally the opposite, actually, from ARP, because ARP's going to bind down at this minus end and add on monomers. But what formin does, it actually binds up at the plus end and then captures another monomer and captures another monomer. and so it's holding on at the top. So this is very different, right, from both ARP and gamma tubulin ring complex. Okay, so here's another one. The ARP complex, formants, and gamma tubulin ring complex are similar in function in that they do what? Okay, we can just raise our hands if we agree. Induce nucleation of actin or tubulin subunits into polymers. Okay, good. Inhibit nucleus formation. Well, that right off the bat should tell you that's wrong. Induce nucleation resulting in filament disassembly. Okay. Expose the NLS, sending subunits to the nucleus. I know. I'm so bad at writing wrong answers, aren't I? I just want to make sure the nucleation thing and nucleus are like as far from each other as possible in your brains. Right, so it's A. Right, so they are all nucleating proteins. They all encourage some sort of growth of actin or tubulin or microtubules. Anyone have a question on that one? Okay. Okay. So we're going to talk now about thymosin. So thymosin, actually, it's just a protein that binds to actin monomers and sequesters them, so they can't be added to growing actin filaments. So the critical concentration for actin, so this is this concentration above which actin spontaneously polymerizes, is about 1 micromolar. So when you have 1 micromolar actin, you're going to get polymerization. But in the cell, actin is present in much, much higher concentrations, like 50 to 200 micromolar, right? So why is this? And thymosin is the answer, and that was actually also a quiz question. Because it's actually kind of hiding that hiding the concentration, and in a way increasing the critical concentration. So what thymosin does is it binds actin and prevents it from associating with actin filaments, and this just makes up a kind of a storage pool of actin monomer that's ready to be mobilized if needed. So when would you need to mobilize some kind of actin monomer? Why would you want a storage pool monomer? What could this, like, if you were a cell, yeah. So you could quickly respond to an external stimulus. Right, so if you have all of your actin busy in some sort of actin filament, and then you need to suddenly move towards something, or you need to prepare for cytokinesis or something that you're doing, you have to then break down your actin and then send it to wherever it needs to be so it can polymerase, right? But if you've got this storage pool of actin monomer, then you just really need to activate it when it's needed. Any questions on that concept? Yes?
You said it creates what was the use of the storage pool again?
Just so it's there when you need it. So your choices in your cell are monomer, polymer, or like actin filament, or wait, those are really your two choices, right? And it's that concentration of free monomer that determines how much is going to be present in filament form. But by having something like thymosin there, it will bind and prevent all that monomer from participating in filament formation and have it there so it's ready to be used. Okay, so if you've got something that's keeping actin monomer inactive, you need something that's going to help it to become active, right? Because, well, if it's just like forever inactive, that's not good for the cell either, is it? What if I just said, you won $1 million? Would anyone, like, it's like dead people in here. Sometimes I'm like, okay, you won $1 million, yay! I just need some life. It's a Monday morning, give me some life. Should we do jumping jacks? Everyone. Okay, We'll start Wednesday's class with some yoga. Okay, right. So there's this wonderful protein called profilin, and it competes with thymosin to bind actin monomers, and this actually encourages addition of monomers to the plus end. And so this is what it looks like. There's your profilin, and it binds to actin, and it binds to the end of actin that would normally bind to the minus end of the actin filament, and so it makes it available to add to the plus end. And when binding happens, you get a conformational change in actin, so it kicks off the profilin. So that way actin added to the filament is available for another actin to come on, right? Because if you think about it, profilin's kind of binding there right now. So you can think about it as there's kind of a balance between binding by thymosin and binding by profilin. And as you activate profilin, you get movement of the actin monomers from the thymosin pool to the filament plus ends. And so this is supposed to show you that you've got, let's see. So thymosin is inhibitory, so it's going to prevent addition. And then profilin is actually going to promote addition. Questions? Okay, so this might make it even make more sense. So formins, remember, what are formins? We just talked about them. What do? They nucleate. So formins, they preferentially add profilin-bound actin to the growing plus end of an actin filament. And so the structure of a formin, they have, so there's your actin, there's your formin-bound right there, and the formin has like these flexible protein linkages that hang off. They're referred to as formin whiskers. They're whiskers, like a cat. That you love formins, just like cat. Who loves cats? Okay, you guys are like, oh my God, what is wrong with her today? I don't know. Too much time in the ER yesterday. Okay, and the whiskers actually bind to profilin. And so you have profilin-bound actin being brought in by the formins. So it's kind of, they're called like a staging area for addition of actin monomers to the growing actin filament. Okay, here's another question for you. Formins bind to profilin. A likely role for this interaction is... Does anyone... I don't feel like reading all that. So I had to write it all. I shouldn't have to read it. B. Okay, so formins are... Yep. To mediate addition of actin monomers to the plus end of the growing actin filament. Right, because we just talked about how profilin binds actin in a state that's ready to be added to the growing actin filament. And so formins have these whiskers that bind it and help to bring it in to be added to the growing actin. Okay, good. Okay, so now we just keep talking about there are these things that bind to monomer, and some encourage addition, some prevent addition. Who cares? Well, let's talk a little bit about how this might function in the movement of the cell, right? So profilin, it actually binds to PIP2, which is a plasma membrane lipid, and helps to localize actin monomers to sites requiring rapid actin polymerization, right? We just talked about how often you'll have actin polymerization at the leading edge. If a cell needs to move towards something, then that actin's going to start polymerizing right at the plasma membrane, at the location to form the leading edge for the cell to move. And so this is one of the ways that it's done. So there'll be some extracellular signal, and profilin will be activated, and it will actually compete with thymosin to bind actin, so it's removing it from that inactive pool. And then the profilin-actin complexes will interact with proline-rich membrane proteins. So that's, you're bringing it to the plasma membrane, and then it will add actin to the plus end of growing actin filaments. So you should ask yourself, what does this have to do with regulating cell movement? Can you guys see it? Can you see it in your mind's eye? right? So as you start to grow your actin filaments at the plasma membrane, you're pushing your plasma membrane out. Okay? Do you see it? Do you feel it? Do you love it? We got like 4 or 5 minutes left. Who's like, oh my God, 4 or 5 minutes? I think it's going to be fun. It's going to be a fun 4 or 5 minutes. Okay, so the critical concentration, I kind of just told you this. The critical concentration of actin in a test tube is less than 1 micromolar. However, in the cell, soluble actin monomer is observed at a concentration of 50 to 200 micromolar. Which of the following are plausible explanations for this discrepancy? Right, B. Proteins like thymosin that bind monomer and prevent association with the plus and minus ends of actin filaments are likely to account for this discrepancy, right? ARP nucleates, so that doesn't make any sense. Severing proteins, doesn't make any sense. So basically, it's something that would be creating a pool of inactive monomer, which the only option is thymosin. Okay, good. Okay, so we've been talking about actin a lot, and I have a feeling that tubulin is feeling left out. So let's talk about tubulin. Stathman serves a similar role with tubulin subunits. So we're going to keep going back and forth between proteins with the same function in actin and tubulin. So Stathman binds tubulin subunits and keeps them inactive also, and prevents addition to microtubule ends. This increases the likelihood that a microtubule will undergo catastrophe. Why? If you bind your tubulin monomers, why does this increase the likelihood that a microtubule will undergo catastrophe? What controls whether or not a microtubule will undergo catastrophe? Or I should say, what is catastrophe? Huh? It loses its GTP cap, but what's the result? What's the result in, what's the effect on the microtubule?
Net
Shrinkage, right, okay. So, how come Stathman increases the likelihood that a microtubule will undergo catastrophe? Well, what controls GTP hydrolysis?
Is it because it essentially lowers the critical concentration, which means that addition is slower and slower?
Yeah, so you are not completely wrong, but you're not giving me a direct answer. But so okay. Does it increase GTP hydrolysis? Well, GTP hydrolysis just happens by addition of monomer to the growing microtubule. And the whole GTP cap thing is just basically because the critical concentration is such that, so the concentration of monomer in the cell is such that so much is added, and there's a lag in hydrolysis that you create this cap. But as the concentration of monomer in the cell starts to go down, addition is slower, and then you have hydrolysis of your GTP, and then you lose your cap, and then you have catastrophe. So Stathman is binding to monomer and preventing addition. So it's almost like when you get to that point where your concentration of monomer is low, because the concentration of monomer available to be added is low. And so that allows for hydrolysis of the GTP to catch up. So you have loss of your GTP cap, and then you have catastrophe. So who followed that? Some people followed it. Who didn't follow it at all? Okay. So it's all about, and this is a really important concept to understand with microtubules. You really need to understand that, we got to go in a minute. You really need to understand that there's a lag between addition of monomer and GTP hydrolysis. And the speed at which monomer is added controls whether or not the hydrolysis is going to catch up. And so when you have a huge pool of monomer, it's going to add quickly, so you'll have a buildup. And as you have a smaller pool of monomer, you will no longer be adding as quickly, and then it'll catch up, and then you'll have hydrolysis and then catastrophe. And Staffman makes it so that the effective concentration, so the concentration of monomer that's able to be added, cannot. It's really low. It's really low because it's all bound up by Staffman. So you get catastrophe. Okay, we'll start here later next time.